Welcome back, everyone. Next up, we have Sigyn Therapeutics. It trades on the OTCQB under the symbol SIGY and creates therapeutic blood purification candidates to overcome clearly defined limitations in healthcare. The company's leadership team has extensive public market and medical technology experience, which includes multiple first-in-industry achievements. Please welcome its CEO, Jim Joyce. Nice to see you, Jim. Welcome.
Nice to see you, Anna. Thank you. Appreciate the introduction. Before I get started, I encourage viewers to review our forward-looking statements. They can be accessed on the presentations button on our corporate website. That website is SigynTherapeutics.com. With that said, as Anna mentioned, we create blood purification technologies to overcome limitations of drug therapies. Let me tell you a little bit about our company. We have a proven team with multiple first-in-industry achievements. Most of my team are my former members of a previous company, Athlon Medical, that I founded, built from a single shareholder startup to over 8,000 shareholders on NASDAQ with a peak market value of $200 million. In regards to first-in-industry achievements, we oversaw the development of the Athlon Hemopurifier. This was the first-in-class device to receive 2 FDA breakthrough device designations and became the first device of its kind to receive emergency use authorization from FDA, Health Canada, and the government of Germany to treat a pandemic virus. Time magazine named this one of the 11 most remarkable advances in healthcare. Reader's Digest named it to its top 10 medical breakthrough list. And again, Time magazine named it as a top 25 invention. In regards to our lead product, it addresses significant unmet needs in global health. And it does so by being able to perform functions that are beyond the reach of drugs. And then behind that, we've created an expansive pipeline of drug enhancement devices, primary focus directed towards oncology. From the standpoint of extracorporeal blood purification products within this industry, we believe our pipeline is as expansive and compelling as anything in industry. Our lead product is called Sigyn Therapy. This is a broad spectrum hemoabsorption technology to treat pathogen-associated inflammatory conditions. And when we say broad spectrum, this device has the ability to address pathogens that initiate inflammatory disorders in concert with knocking down inflammatory cytokines and other factors associated with a dysregulated response that can lead to organ failure and death. In terms of these capabilities, we don't believe there's any other public company that has a product with these capabilities. To tell you a little bit about the history of hemoabsorption devices, the field was pioneered by our colleagues at Cytosorbents. They trade on NASDAQ, CTSO. Their product is the Cytosorb device. This is a good product, a really good management team. I encourage you to investigate Cytosorbents more. I can tell you I spent 5 years with their team in a Department of Defense DARPA program looking to develop blood purification technologies to treat sepsis in wounded warfighters. Their device incorporates an adsorbent component that depletes targets below 5 nanometers in size. And as they reference, it has 45,000 square meters of surface area to absorb their targets. And as it relates to Sigyn Therapy, we incorporate multiple adsorbent components. We have approximately 200,000 square meters of adsorption area. To put that into context, that's about 50 acres of adsorption area to remove our targets. Our device is highly efficient, approximately 15 passes of the entire bloodstream through the device during a 4-hour period. And again, this is very dependent on the size of the patient being treated. And we're able to remove therapeutic targets up to 200 nanometers in size. This is why we can address both pathogen sources of inflammation as well as the inflammatory factors themselves. Additionally, our device is designed for use on the global infrastructure of dialysis machines already located in hospitals and clinics worldwide. This is an image of our device being deployed in action during animal studies at University of Michigan. And again, in this image, it's being deployed on a Fresenius dialysis machine. So when we envisioned this product, we envisioned a device that could address both pathogen sources of inflammation, again, in concert with damping down a dysregulated immune response. A number of years ago, I was invited to testify before Congress on the use of medical devices as countermeasures to address emerging pandemic threats. During my testimony, I pointed out there would be a need for a device that could address a broad spectrum of pathogens, specifically viral pathogens, and that there would also be a need for a device that could address the cytokine storm, the excess inflammatory response to infection, which often leads to death in infected individuals. Again, Sigyn Therapy performs both of those functions, and we have completed studies that demonstrate that through blood plasma validation studies, these are called in vitro studies, the ability to address a broad spectrum of inflammatory cytokines, bacterial toxins, including gram-positive and gram-negative bacterial toxins, including endotoxin, viral pathogens, including COVID-19, hepatic toxins, and cytovesicles. This was a model study looking at extracellular vesicles that have the capabilities to transport inflammatory cytokines throughout the circulatory system. These cytokines are not just circulating freely in the bloodstream. So as a result of these capabilities, we're a candidate to address significant unmet needs in global health. And this includes sepsis, which is a leading cause of hospital deaths around the world, endotoxemia, which is a leading trigger for causing sepsis, pandemic viruses whose occurrence is increasingly happening as a result of intercontinental travel, global warming, and urban crowding. The quiet pandemic that keeps building up is drug-resistant bacterial infections, and then community-acquired pneumonia, which is one of the leading causes of infectious disease deaths. Our next step, based on previous studies, is an IDE submission to FDA related to first-in-human studies. IDE stands for an investigational device exemption. We call this a modular study strategy, and the objective is to enroll a patient group to demonstrate safety of our technology and then use that safety study in a modular manner to bridge into pivotal studies required for approval. So we plan to, and by the way, to apply for pivotal studies for approval of a variety of different indications. The goal is not to have to run a safety study over for each different indication. So in this study, we plan to enroll 12 to 15 dialysis-dependent end-stage renal disease patients with endotoxemia and concurrent inflammation. A high percentage of these patients suffer from endotoxemia, which leads to inflammation. And these patients, once they're on dialysis, these factors contribute to early mortality. Once they're dialysis dependent, their lifespan is typically 60 to 84 months. There's approximately 550,000 ESRD patients in the United States that receive 85 million dialysis treatments each year. The incidence of endotoxemia alone is 40 to 90%. The study protocol, we've developed this protocol in collaboration with one of the leaders in the dialysis industry. Sigyn therapy will be administered in series with regularly scheduled dialysis treatments. So nothing changes for the patient's treatment schedule, which is typically 3 times a week, 4 hours of treatment. They'll receive 6 total treatments as defined in the protocol. The first 3 treatments are their normal dialysis treatments, followed by 3 dialysis plus Sigyn therapy treatments, and Sigyn therapy be put in series with their dialysis cartridge. This allows us to have the patient to be their own control, where we can look at blood chemistry, what happens during normal dialysis without Sigyn therapy in series, and then looking at dialysis with Sigyn therapy in series. Primary objective, again, safety, but we also get to quantify endotoxin and inflammatory cytokine reduction. Something we set forth as a parameter for developing devices, again, we don't license devices or acquire technologies. we develop all of our technologies. And as a criteria, if proven effective, our technologies need to provide a competitive advantage within an established industry space. And within the dialysis industry, we think that's the case. There's two leading players in North America, Fresenius Medical Care and DaVita Kidney Health. They dominate this market. And what we did is we projected the ability or the revenue generation that would occur if we could extend the life of their patient population by 1 month, what the impact would be from a revenue standpoint. So as it relates to DaVita, they have almost 200,000 ESRD patients in their network. In North America last year, they generated $11.6 billion in revenues. And by our calculation, for each month you could extend the life of their patient population, that would add about $967 million in revenues. That's not inclusive of revenues that would be generated by reduced hospitalization. These ESRD patients, they spend quite a bit of time in the hospital each year. And when they're in the hospital, the dialysis companies lose revenue. They're outside of their network when that happens. So that would add an additional $240 million in revenues for each week of reduced hospitalization. And that does not include revenues related to Sigyn therapy sales, both to ESRD and non-ESRD markets. And again, this strategy has a potential opportunity to put the dialysis companies in the ICU to treat other conditions beyond their dialysis patients. As it relates to Fresenius, they generated $14.5 billion in revenues last year. They have about 208,000 patients in their network. Based on our calculation, we project for each month of extended life of their patient population, that would be worth $1.2 billion in additional revenues, about $300 million for each week of reduced hospitalizations. And again, that's not inclusive of opportunities to generate revenue through Sigyn therapy and sales to non-ESRD markets. So now I want to talk to you about our drug enhancement pipeline. We think this is a novel approach where devices can actually be designed in advance to augment the benefit of drug therapies, specifically to improve their delivery and reduce their toxicity. In October of last year, we filed a patent called Systems and Methods to Enhance Chemotherapy Delivery and Reduce Toxicity. This is related to a system where there's two products that comprise the system, ChemoPrep and ChemoPure. First, I want to tell you a little bit about the limitations of chemotherapy. First, we know about toxicity. There are high rates of chemoresistance that occur in patient populations, but perhaps the most shocking number is that the actual tumor site delivery of chemotherapy is typically between 0.5% to 3.5% of the drug actually gets to their target tumor site, which we think that's just not acceptable. We think we can improve upon that. So with ChemoPrep, it's a system designed to be administered prior to the infusion of chemotherapy. Our goal is to improve delivery or maintain the same delivery with half a dose of chemotherapy. And by using a half a dose, that right off the top is a 50% toxicity reduction. And our strategy really corresponds with a trend to de-escalate cancer treatments, looking for approaches to cut back on therapies to improve quality of life, but without hurting the odds of survival. On a more urgent basis is the fact that chemotherapeutic drugs, there's a significant shortage and they're being rationed in their patients. So the ability to reduce the dosage required, but yet increase or maintain the tumor site saturation could be of critical importance. And this is an ongoing issue, not just right now. It's been an issue in the past, and it's expected to be an issue going forward in the future. So again, ChemoPrep, we administer this prior to the infusion of chemotherapy. Again, goal, improve delivery with a half a dose, which right off the top reduces toxicity by 50%. So how we do this is we're targeting a circulating factor called cancer exosomes. And as pointed out in this paper authored by Raghu Kalari at MD Anderson Cancer Center in Houston, the blood of cancer patients is estimated to contain about 4,000 trillion exosomes. That's 4 quadrillion. And the circulating presence of these particles corresponds with drug resistance, with chemo resistance. So removing these, we believe, would not only enhance the delivery, but would reduce chemo resistance. And just to have you understand the magnitude of circulating tumor X's, based on an average size of 100 nanometers, that's 248,000 miles of these particles that can cause chemotherapy interference. And in regards to the interference, I want to be a little more specific on what they do. Tumor X's block and sequester chemotherapy delivery before it can be delivered to the target tumor site. They can even export chemotherapy once it's been delivered from the target tumor site. And again, as mentioned, they're a prime contributor to treatment resistance. As I mentioned, this is part of a system, chemo prep before the administration of chemotherapy, and chemo pure to be administered after chemotherapy to further reduce toxicity related to off-target chemo. At a certain point in time after the delivery of chemo, circulating chemo is more likely to cause toxicity in patients than it is patient benefit. So additionally, removing that off-target chemotherapy could be very beneficial to patients. And in regards to some early data, the primary methodology of delivering chemotherapy is through nanoparticles called liposomes. And this was just looking at our adsorbent components that we'd be using to remove these liposomes that deliver chemotherapy. And we had a pretty substantial impact in vitro preclinical studies. And it's important to recognize that historically in exosome research, liposomes are also used as a model system because of their size and characteristics for exosome removal. So we think we're on the right track here right out of the blocks. Next target I'm going to talk about to enhance the benefit of drug therapies is a product called Immune Prep. And this is related to a patent that we filed in May called Devices for Enhancing the Activity of Therapeutic Antibodies. This is basically immuno-oncology, immunotherapeutic antibodies, including checkpoint inhibitors. Now, the limitation of therapeutic antibodies, again, poor delivery, less than 5% are delivered to their intended target. And in this case, the drug decoys are a little more expansive. The immune system will create anti-drug antibodies that express the antigen. That's the target binding site of the antibody. There's also circulating soluble proteins that are produced in circulation that express the antigen binding target of the antibody. And again, cancer exosomes or tumor X's, they are produced and will express the antigen binding target and interfere and decoy the drug from delivery. And a good example of that is with rituximab in the treatment of lymphoma patients. In this study, it was reported that half of the rituximab was found fixed to exosomes 3 hours after the rituximab infusion. Now, rituximab has a half-life of 72 to 76 hours. So seeing that type of adsorption or binding just 3 hours in is pretty substantial. So our solution is Immune Prep, and this is a reverse decoy mechanism. So Immune Prep incorporates an antibody that's actually going to be infused into the patient. It's the same antibody, and we pre-treat to deplete circulating decoys prior to the antibody infusion. So anything that binds to the antibody inside of Immune Prep as the circulatory system is passed through would have had the ability to block the delivery of the infused therapeutic antibody. And decoy depletion increases antibody availability to bind their intended therapeutic targets. And we think this is a de-risk strategy because it allows us to incorporate market-approved antibodies that have already been demonstrated to be safe and effective in humans, and antibodies for which scalable production is established. And the market opportunity is pretty significant. Last year, there were 162 market-approved antibodies that generated almost $200 billion in revenues. And there's 1,200 therapeutic antibodies in the clinical pipeline right now. So, as I said from the outset, at Sigyn Therapeutics, we create blood purification devices to overcome limitations of drug therapies. We have a proven team with multiple first industry achievements. Our lead product addresses significant unmet needs in global health, and we have an expansive pipeline of devices to augment the benefit of drug therapies, specifically oncology related at the outset, but the intent to improve the delivery of drugs and to reduce their toxicity. So near-term inflection points for the company, our IDE submission to FDA, critical inflection point. The disclosure of our clinical site locations. At present, we've identified 5 sites that we're evaluating. The expansion of our management team, collaborative relationships, and reporting on studies underlying the advancement of our pipeline therapies. So with that, I appreciate your time. I encourage you to reach out to me, especially by email. My email is jj@Sigyn Therapeutics. I have over 20 years of public company CEO experience, board leadership experience, and what I recognize is, a couple years from now, when I look at our top 20 shareholders, there'll be people that are passionate about what we're doing and have a relationship with management. So I encourage you to reach out and communicate with me. With that said, I'll open up the floor for questions.
Great job, Jim. Yes, we do have some questions for you. Let's start with Athlon Medical. It seems like you had a lot of success there. So why did you leave?
Oh, well, I didn't leave. About a week after receiving our second breakthrough device designation award from FDA, which opened up the opportunity for us into oncology, the board decided they wanted to replace me as CEO. So that was at the very end of 2018. And unfortunately for myself, my family, and shareholders, the value of the company depreciated by a little over 96% the next year. But as a result, we're now advancing new technologies with new strategies that are expansive, and we really think we can achieve our mission of creating products that save lives.
Perfect. You also mentioned the importance of creating devices that offer a strategic competitive advantage within an established industry. So talk a little bit about why that's so important.
Anna, I think that's critical for success in our industry. Our space, extracorporeal blood purification, is not a large industry. There's always been tremendous potential for this to become a significant industry segment. But historically, many of the devices created, they address indications for which there's no established market. So if the product was to get approved, the company then has to establish the market for the device. And when we established Sigyn Therapeutics, it became clear to us that we need to create technologies that will create a competitive advantage inside of an established healthcare segment. As an example, what we're doing with dialysis patients, there's opportunities that are larger market opportunities with Sigyn therapy. But we recognize that in the dialysis space, if we can improve the quality of life, extend the life of these dialysis patients, who are suffering from endotoxemia, from a chronic basis, which leads to inflammation, which increases cardiovascular disease, that will create a competitive advantage in that space if demonstrated to be effective. And the same is true when we designed immune prep, when we designed chemo prep and chemo pure. We recognize that if we execute on our strategy, these will be important technologies to the drug industry. And I wouldn't want to be the drug company that doesn't have access to them or doesn't own them. So it's an important aspect of our strategy going forward.
Perfect. Ross Lucera wants you to clarify the IDE. How does this affect the timeline of the studies?
Yeah, the IDE, this is really the submission related to our first in human studies of Sigyn therapy. The patient population will be 12 to 15 patients. The timelines, we can't tell you exactly when FDA will approve the IDE and give us permission to initiate our studies. My experience is that our reviewers are at CDRH, which is the medical device division of FDA. My experience is that they don't give you a pass, on your first submission. Usually you're going to have to sit down and discuss certain points. There's going to be questions, and there's going to be a turn of the IDE or two in discussion. So, our hope is we're up and running in the next year. And I mentioned 5, clinical sites that we're evaluating, 12 to 15 subjects and we're enrolling subjects that there's a very high incidence in the patient population. So once we're up and running, we hope to be very efficient at bringing this to conclusion. And then the key thing is it's a modular strategy. So with devices, this is a class 3 medical device. You have feasibility study, which is safety, and then a pivotal study for approval to demonstrate efficacy. We want to leverage a modular strategy where we move into pivotal studies. We complete the safety study and move into pivotal studies, potentially of community-acquired pneumonia, or if there's an emerging pandemic threat, or sepsis. But, we want to be running expansive studies, but the lead indication, end-stage renal disease. And in terms of timing and some of these things, you just can't control everything in terms of the process with FDA, has been my experience. But I think we've developed a very strong regulatory submission that we've drafted, and have done so based on our years of experience in interacting with FDA.
We have a few questions about ChemoPure. John Clements wants to know, can these be used as a substitute to any shortage in chemo?
We would envision that this device would allow, when there is shortages, would allow for lower doses of chemo to be administered without losing the effectiveness of the drug. That's our goal. Lower doses that reduces toxicity right off the bat, but by removing circulating tumor exosomes that play a significant role in blocking the delivery of chemotherapy, we think we can maintain or increase the actual delivery. And if we can demonstrate that, I think we could play a very important role. And the design of our system that I didn't mention earlier is we've designed it to work on blood processing systems that can easily be placed in the infusion centers where patients are receiving their chemotherapy, the same with therapeutic antibodies, so that there's a convenience factor. The patient doesn't have to go one place or another. These are systems that we're working to advance that even potentially could use the same port that is going to be delivering the drug agent. So we're trying to make this convenient and highly something that could be, acceptable to oncologists.
And Ursula Bird wants you to give a timeline on your near-term inflection points.
Well, there's several of them. We're constantly, the nice thing about having a pipeline is we should have a nice flow of early data from our pipeline products. In terms of, I mentioned expansion of management, we've been in discussions with different individuals that we think could be a very good asset to our team. Related to our IDE submission, we are finalizing the protocol with a collaborator in the dialysis industry. And I think we will probably close in on finalizing that protocol, pick some clinical sites, and then get our IDE submission into FDA by year end. Those are the real key timelines. And again, I mentioned collaborative relationships. I think that, once we build a little bit more momentum. We've been very focused and quiet over the last year. I think we can establish, a number of very important collaborative relationships that could provide additional credibility to what we're doing. While we were at Athlon, we established collaborations with more than 20 leading government and non-government institutes that got involved with what we were doing and helped advance our studies. And I think we can do the same thing here.
And Don Nicholson asks about the quality of the product is one of the most critical aspects, obviously. So what measures are you taking to ensure top quality? Good question, Don.
And nice to hear from you. So we designed our product around components that are already available. So at the core of our product with Sigyn Therapy, we incorporate our adsorbent components within plasmapheresis devices that are already in the marketplace and used in providing plasmapheresis treatments, separating plasma, from patients for plasma donation, or other procedures. But we focus on modifying products that are already scaled and in production. We think that is a critical aspect of quality control. And it also is an important aspect to our regulatory advancements, because FDA really appreciates that you're incorporating components that perhaps they've already seen before, or perhaps they've approved for other types of indications. So we don't, from an R&D standpoint, we try to keep our parameters such where what is FDA seen, what components are available, how do we reconfigure those for our use, which means we don't have to fully recreate new things that require, a lot of excess development.
Well, perfect. Jim, do you have any closing remarks for our viewers today?
No, I appreciate, all of you that's participated. I think we've put a really good foundation in place for the company and are really looking to start building value for our shareholders. And again, I encourage you, if you'd like to interact with the company, please send me an email. Letter is jj@sigyntherapeutics.com. But again, Anna, thank you, and I appreciate everybody's time today.
Perfect. Thank you so much for joining us. We look forward to having you back with some updates.
All right. Take care.
Okay, everyone, stay with us. We'll be back at 2:40 Eastern with Dinah Cert. So we'll see you in a second.